All right, good afternoon, everyone. Welcome to the 28th Annual George A. Miller Prize Lecture. I'm Allie Cohen, and I'm a postdoc at NYU and incoming assistant professor at Emory. And it's my great honor to introduce the lecture and this evening's speaker. The George A. Miller Prize in Cognitive Neuroscience was established in 1995 by the Cognitive Neuroscience Society to honor the innovative scholarship of George A. Miller, whose many theoretical advances have greatly influenced the discipline of cognitive neuroscience. The first 10 years of the prize were funded by generous support from the James S. McDonnell Foundation, and it is now supported by the Society. The prize is awarded to a scholar whose contributions are characterized by distinguished and sustained scholarship and research at the cutting edge of their discipline, and that has in the past or has the potential to in the future revolutionize cognitive neuroscience. Today, I have the enormous privilege to introduce the 2022 recipient of the George A. Miller Prize, Dr. B.J. Casey. Dr. Casey is a professor of psychology at Yale University and a faculty member of the Justice Collaboratory at the Yale Law School and of the Interdepartmental Neuroscience Program at the Yale School of Medicine. Professor Casey pioneered the use of fMRI to study the developing human brain, particularly during adolescence. Her work substantially contributed to the foundation of what has become the flourishing field of developmental cognitive neuroscience. Dr. Casey's scientific discoveries have been published in over 220 articles in top-tier journals, including Nature Medicine, Nature Neuroscience, Neuron, PNAS, and Science. Her work has been cited over 60,000 times and highlighted by many media outlets such as NPR, PBS, The New York Times, and National Geographic. She has received numerous honors and awards, so I won't list all of them, but these include the American Psychological Association Distinguished Scientific Contributions Award, the Association for Psychological Science Lifetime Achievement Mentor Award, and she's an elected member of the American Academy of Arts and Sciences. Dr. Casey has served on several scientific advisory boards and panels, including the NIMH Board of Scientific Counselors and NIMH Advisory Council, and the National Research Council of the National Academy of Sciences Committees on Assessing Juvenile Justice Reform, the Science of Adolescent Risk-Taking, and Sports-Related Concussions in Youth. Her work has not only been transformative within the field of cognitive neuroscience, but has also had tremendous real-world impact, particularly in the legal realm. Dr. Casey has been cited in amicus briefs on the sentencing of young offenders for the U.S. Supreme Court, and she's presented adolescent brain science to congressional staff on Capitol Hill, to federal judges, and state Supreme Courts. Dr. Casey is as passionate and impactful in her mentoring as she is in her science. In fact, it isn't an exaggeration to say that every developmental cognitive neuroscientist can be linked through 6 degrees of B.J. Casey, usually less. As a proud first-degree link, I've always marveled at the profound effect B.J. has had on the careers of cognitive neuroscientists, both those of us who are lucky enough to be her trainees and those who have entered her orbit through the many institutes and training programs that she's created and contributed to throughout her career. She's a strong advocate for early career scientists and frequently spotlights her trainees, collaborators, and other scientists in our field, and I'm sure we'll see some of this today. I'm so grateful that BJ was my PhD advisor and that I know I can always count on her for feedback, guidance, and unwavering support. Through both her innovative research and her exceptional mentorship, B.J. Casey has truly revolutionized the field of cognitive neuroscience and embodies the ideals of the George A. Miller Prize. Before I present this well-deserved award, a brief reminder that poster session C follows this talk and will be in the Pacific Concourse from 5 to 7 p.m. It is an honor to have Professor Casey with us tonight to present the George A. Miller Lecture entitled Cognitive Neuroscience in the Age of Discovery. Please help me welcome Dr. B.J. Casey.
I don't think it gets much better than to have one of your former students introduce you. But I hope it's not only going to go down from here after all the great things that you said. It's such a privilege to be here, to see so many of my colleagues in person, and such an honor to receive this award. I was surprised and totally humbled in receiving it. So I really just want to thank the Society and the Awards Committee for giving me this honor. I always like to begin by acknowledging so many that have influenced my research, and in fact, this award reflects their work as much or more than my own. And I've really had the opportunity to work with so many brilliant scientists, and I'll be highlighting them in a blue border so you'll know who are trainees or indirectly have been mentored in the lab versus others whose work I just absolutely love. I also want to acknowledge Mike Posner. He took a chance on me early in my career, and he placed me in a developmental cognitive neuroscience institute that really built the foundation for the work that I'll present today. And Mike, I would not even have been on his mark if it weren't for the patience with and challengings of me by my graduate and postdoctoral advisors, John Richards, who's here. Hi, John. And Randy Engel, and also Judy Rappaport. So I just want to take a few moments and unpack what I mean by this title. So in terms of age of discovery, I'm talking about an age of discovery of our field as we're seeing these advances in imaging acquisitions, new analytic tools. We're even beginning to be able to look at microstructure with restriction spectrum imaging. We're able to begin to combine techniques where we can look at microfunction with laminar fMRI. And we also have novel animal models too, all of which are informing one another to help us make more discoveries in the field. But it's also an age of discovery in terms of, I think of the whole life course as an age of discovery. And we're learning the limits and the demands on us at every stage, but particularly during adolescence, at a time when you no longer really have the buffer of the caregiver, and you're learning to explore and understand the boundaries in order to become a sufficient and viable member of society. I would argue this is one of the more particularly special ages of discovery. So I'll try to unpack this in the context of three questions and briefly touch on each. And one is, how has cognitive neuroscience changed in the 21st century? And I'll just give you some examples from our own lab. How cognitive neuroscience has informed our understanding of the developing brain and mind, and also how it's informed the treatment of the developing brain and mind. So if you'll bear with me, let's just stroll down memory lane. And I want to show you the first study that we published using this wonderful technology of functional magnetic resonance to look inside the behaving brain. And that first study used an in-back task, one that was described by Danny Bassett in just a brilliant talk yesterday on cognitive effort. And here we were comparing of a high memory load to a low memory load, and looking at patterns of brain activity, and we see areas of the dorsolateral prefrontal cortex that were activated. We even compared this pattern to adults, although we had no adult sample in the study, really large sample of 6. We then went on to do reproducibility. We were bold. And across 4 sites, again, you'll see these large samples. We showed that we could replicate this general pattern in the prefrontal cortex. And again, we didn't even do a simple conjunction analysis to see if it overlaid in the same regions. But now, with the advent of big data, we have opportunities to look at more representative samples, larger samples, and they bring many opportunities. In fact, they help to democratize access to imaging data, because we don't have access to these facilities all around the world, and imaging can be expensive too. And so there's a beautiful paper by Monica Rosenberg where she actually talks about this wonderful balance between prediction with these big data and also explanatory experiments, and how they really need to go hand in hand. And highlights not only democratizing data access, but the reproducibility, replication, and hopefully that our discoveries will inform policy. Now, this map keeps growing. The blue areas that you see are studies that have at least 500 children with imaging data, and they're also phenotyping them as well. And one of these studies is the Adolescent Brain Cognitive Development, or ABCD study, that I've been heavily involved in. Many of you are familiar with this study, but basically, we are following approximately 12,000 9 and 10-year-olds over 10 years in 21 sites in the United States. And in addition to getting structural and functional imaging, we're also getting various rich phenotypic data as well. And with data sets like this, we can begin to ask questions that go beyond the ones that I showed you that were occurring in the early 1990s. And again, this is work that I want to highlight by Monica Rosenberg, Stephen Martinez, Cristina Rapuano, and several others, where she looked at the ABCD version of the NBAC task that was performed in the scanner, and then looked at the contrast of the high memory load to the low memory load, and then wanted to see how that neural signature and brain pattern related to performance of a completely different working memory task that was performed outside the scanner. So the list sorting task, where you are given a list of items and you have to give them back in the order of the size that they are. And so what we were able to show is that you see this nice frontal parietal network and also a dorsal attentional network that's activated when you compare load, high load to low load. But if you look at other contrast in that task, they don't relate to performance on the list sorting working memory task. So this is just looking at the different stimulus categories. There were emotional, positive, negative, and neutral, and also places. But you don't see that positively relating to working memory ability as measured by the list sorting task. And two other tasks that we have in the ABCD, or the monetary incentive delay task and the stop signal. And likewise, we don't see this positive related working memory ability specifically as measured by the list sorting task relating to that pattern of behavior. I must say, John Duncan's session earlier today was just fabulous, and he was showing the frontal parietal network, how the data almost look too good to be true when you run an NBAC that's either visual or auditory, which really sort of captures how what we're capturing here. You can also look at reproducibility. This is just looking at the first release of the ABCD data with 9 and 10-year-olds, and then the second release of the 9 and 10-year-olds, and you see the exact same pattern. And then what we've done recently, Cristina Rapuano, you can catch her poster, I believe it's on Tuesday. We developed this open access accelerated adult equivalent of the ABCD study. Instead of scanning these adults every 2 years for 10 years, we're actually scanning them. We have scanned them 5 times, but they were separated with a week apart. And what you can see from the graph here is that for the NBAC task, we're not seeing practice effects, even in that short period of time. And when we look at within subject overlap, we are again seeing these frontal parietal and dorsal attentional networks for the NBAC task pop. And as no surprise, we're seeing greater similarity within than between subjects in this pattern. So the data that are available include the structural, diffusion weighted, and all the resting state too. And again, at the poster, you can use this code or at the talk and pull that. Now, some of you may have attended Lena Scalaband's poster this morning. Lena's been looking at the ABCD working memory NBAC task data. And what she noted was that there is an advantage with having faces as stimuli in the working memory task. But then when we do the surprise subsequent recognition test, you no longer see that advantage. In fact, it looks more like we're seeing better recognition memory for the places. Now, in part, that might be because we have 3 categories of faces and only 1 category of places. And so Lena wanted to try to explore this more, and using representational similarity analyses, look at areas that are sensitive to categories, and also control visual areas like V1. So here's the fusiform, the parahippocampal gyrus. And then to look at a hypothetical model which proposes that the more similar you are in your representation between faces and dissimilar from places, she was hypothesizing would be associated with that better memory for places that I'm showing you to the left. But that we might see interference associated with similar representation of different phase categories that might lead to a negative correlation in performance. And this is just showing you the representation of categories or scene. You see more similar representations among faces, between faces, than faces and places. And you don't see that in V1. And we see exactly what Lena predicted, that you remember items from the MBAC task, those places, the greater the similarity in representation among faces and dissimilarity from places. And with faces, we don't see any evidence of interference, but we see this novel finding of similarity in representation of faces seems to be related to your ability to recognize a novel face that you haven't seen before. So track down Lena and get her to talk about her hypotheses for these results. Just want to share one more study where we're taking advantage of the ABCD study, and I'll have to say during the pandemic, I had a new whole appreciation for open access data for my graduate students and junior faculty. So Erica Bush is a graduate student, and she's been really trying to take advantage of the large sample of twins that we have in the ABCD study. And she's very interested in understanding how functional connectome granularity relates to estimates of heritability, reliability, and prediction of cognitive ability. And let me just back up. So basically, she's first looking at anatomically aligned and hyperalignment, both at the coarse grain level of functional connectivity, which would be parcel to parcel or region to region, and then fine grained, which is vertex to parcel. And what she shows in youth is similar to what's been seen in adults, but she sees higher reliability in this hyperaligned, fine-grained functional connectivity. But when she looked at estimates of heritability, it's the anatomically aligned and coarse-grained functional connectivity that's more related with heritability. And the functional significance of this is in looking at which of these is really more predictive of cognitive ability, and using a principal component analysis and identifying a composite score for learning and memory, which included the list sorting task I've mentioned before, she shows that it is the hyper-aligned, fine-grained connectivity that best predicts cognitive ability. So she's been thinking about this in the context of a Lego theory, so that the coarse connectome may provide this heritable scaffolding for a more individualized, experience-based, reliable connectome. And it's like a Lego set in that you can be given specific Lego pieces, but they're parameterized in terms of what you can do with those Legos. But if you give the identical items and pieces to two children, you're going to get unique creations by both of those children. So let me just finish up this big data section by highlighting that anytime we're using other individuals' data, that we recognize the limitations and we recognize the harm that can be done. So remembering that with developmental samples, this development is occurring within a broader social context, some of which has been measured, some of which has not, and that the potential for change is there for all youth. And so using deterministic language or harmful narratives, we really need to make sure that we're responsible and don't use those so that we don't disproportionately impact marginalized youth. I want to switch now to the second question of how has cognitive neuroscience informed our understanding of the developing brain and mind. And I just want to go ahead and tell you what my punchline is from this. And that is, we can no longer think of the developing brain as just this little adult brain. And that all we see is linear growth, right? There's a lot of evidence that's showing nonlinear changes and dynamic changes over the lifespan, but particularly during this early period of development that we've been investigating. Okay, so we were talking about regional changes in the brain, and so basically what we're seeing is sensory motor areas are changing and reaching that peak cortical thickness and subsequent thinning earlier than association areas like the prefrontal cortex shown here. But development isn't all about the cortex, right? And we think about adolescence, often we're thinking about emotional, maybe more primitive responses that they might show from time to time. So I always like to show this wonderful study from Terry Jernigan's lab when Elizabeth Sowell was a graduate student. And very early on, she was showing that, yes, there are these changes shown in fuchsia, the prefrontal cortex, but we're also seeing subtle changes in subcortical structures that are very important and have been related to reward and emotional learning. And Sarah Jane Blakemore has even shown that you see only subtle differences in the amygdala and the ventral striatum over this period of adolescence, but more dramatic changes, which she illustrated last night in regions of the prefrontal cortex. Now, these were structural changes. With the advent of functional MRI, we've been able to, as adults, voyeuristically look inside the behaving teen brain. And I like to show this slide, and this is for a lot of graduate students and trainees. This is my son who got into nature before I did. And they are highlighting an article that one of my graduate students published in Journal of Neuroscience, which they rejected the year before because it was not of sufficient interest to their audience. So never, right? It's like you'll get in there in different ways, or your child will, yeah. So the graduate student happened to be Adriana Galvan, and we were having a lot of discussions about how people were thinking about the prefrontal cortex and adolescent behavior. And it was adolescents do what they do because they have an immature prefrontal cortex. And Adriana and I were like, well, children have an even less mature prefrontal cortex, and they don't do some of the things that our adolescents are doing. So she was trying to think about ways in which we could try to capture this and think about systems that we could try to measure that might be related to risk-taking behavior. And so the first thing that came to mind was trying to think about how young people process rewards, and is it different over development? And using a paradigm that built on manipulating, reward predictions, but manipulating the probability of a large or small reward and of the frequency of rewards, she showed that adolescents have an exaggerated response in the ventral striatum in receipt of large rewards relative to small rewards, a pattern not seen in children and adults. And while there are changes also in the orbital frontal cortex with regard to that contrast of large versus small reward, you don't see anything adolescent-specific in this pattern. She then went on to show that the magnitude of the response in the ventral striatum was associated with risk-taking behavior. So individuals endorsing their engagement in leaving a party with a stranger or engaging in sports that are risky, like rock climbing. And the adolescent-specific pattern that I showed you in the ventral striatum has been shown in many labs across the globe. And I just want to highlight one by Evelyn Crone and Anna, where it's longitudinal data, 3 time points for over 250 individuals, and they still show this inverted pattern. Now, once you see sort of this exaggerated response to rewards or gains versus losses, the question is, how is that biasing or related to behavior beyond just asking a simple survey to your youth to see if they're engaging in risky behaviors? So we wanted to see how adolescents respond to cues that are predictive of positive outcomes, such as positive smiling faces, based on stimuli developed by Nim Tottenham, who's at Columbia. And this experiment was led by Leah Somerville, where what she showed was that you see another adolescent-specific pattern in response to these positive social cues in the ventral striatum. And again, you don't see this in children and adolescents. And importantly, she was able to show that this was related to the adolescent-specific pattern in behavior. So teens were far more impulsive to these positive social cues than they were to neutral. And again, children and adults weren't showing that pattern. Now, again, This was a go, no-go task that we were having them perform, and we know from animal and human imaging studies that the right inferior frontal gyrus, or ventrolateral prefrontal cortex, is an area that comes up time and time again that's been implicated in response inhibition. And what we see is elevated activity in children and adolescents when they're performing that task well, so actually successful inhibitions, and more activity from adults. But again, this isn't an adolescent-specific pattern. It's more showing two different patterns, and we think it's really about how these regions are going to be talking to one another. I want to take it one more level. Like, you can stop responding to a cue or a cupcake that I saw out there, but we can use cognitive strategies to try to prevent us from doing that. My strategy was simply giving a talk in which hopefully there's a cupcake I can have later. And this is work that was with the late Walter Mischel and also with Kevin Oxner. And I have to say, it was just really led tour de force by Jen Silvers when she was a graduate student at the time. And so the question really is, how does cognitive reappraisal of appetitive cues change throughout adolescence? And what she showed is with age, you actually see a decrease in craving of information, whether you imagine it close or you reappraise it to think of it as being far, so less enticing. And when she looked at the pattern of brain activity, looking at whether or not you're imagining this close or far, or simply looking at a fixation, what she showed is that older age was predicting less activation in the ventral striatum, the same area that we've been talking about. And again, this frontal parietal network that we were talking about earlier, you're seeing increases in activity in this region in age as they reappraise. Now, so far we've been talking about appetitive items and cues, but adolescence isn't all fun and games, as is life. And so Todd Hare, when he was a graduate student, he really wanted to understand emotional reactivity and regulation to cues of potential threat, or fear cues, fearful faces. And we were surprised to see this exaggerated response and more variable response during adolescence to repeated presentation of fearful cues. Again, a pattern not seen in the younger or the older ages. But I think really a unique finding from this study is sometimes you stop at the magnitude, but he wanted to explore, with repeated presentations of fearful expression. If I was afraid right now, you might look behind you or in the back of the room to see if there was some threat there, right? But with repeated presentations and nothing's happening, it's like, get over yourself, all right already. Like, let's just bring that down. So he showed that your change in the activation of the amygdala with repeated exposures over time was associated with endorsement of high anxiety. So these are negative scores, and they're negative scores because early on we just took this simple subtraction of the magnitude of the activity at the first half of the experiment and then in the second half of the experiment, and did it decrease over time? And if it's a negative score, it means that it was sensitized or increased in time. And then he went on to try to interrogate that and see what's associated with this habituation of this activity. And it was inverse coupling of the ventromedial prefrontal cortex and amygdala activity that was associated with decreases in amygdala activity with repeated exposures. And this inverse coupling tends to be a switch that shows up around adolescence, and Dylan G. and Nem Tottenham have both sort of shown this pattern as well. Well, now let's think about how we can cognitively reappraise emotional stimuli as opposed to just not responding to them or getting over ourselves when we realize nothing bad is happening. And so this is cognitive reappraisal, again, tour de force by Jen Silvers and also a former graduate student with me, Chelsea Hellion. And so how does cognitive appraisal to aversive or negative cues change throughout adolescence? And again, you're basically seeing that we get better at reappraising as opposed to reacting with age. And these changes continue beyond 18, 19, and even 20. And if you look at the neural correlates, or what is simultaneously corresponding with this reappraisal, is age predicts that less amygdala activity during reappraisal. And activity in ventrolateral prefrontal cortex during this task appears to mediate that association of age predicting a decrease in amygdala with age. But the really cool finding that sort of had me up at night trying to understand was this finding of the correlation between the ventrolateral prefrontal cortex and amygdala during cognitive appraisal was actually moderated by the extent to which the ventromedial prefrontal cortex and amygdala were inversely coupling, just as I had shown you before in terms of regulating individuals who have low anxiety and are regulating that. So it began to make me think that maybe what we're seeing, or maybe what's happening, is we need development of cortical subcortical circuits and connectivity to actually instantiate and facilitate connectivity of more other cortical circuits interacting with one another. And so this has made us think more carefully about our simplistic imbalance model that Adriana Galvan and I developed early on based on her reward work and looking at changes in circuitry in those regions that seem to be involved in desire and fear and rage before I think my talk's on iCloud. My apologies. Yeah. So let me just go. Any questions? Hopefully I can get through this quick. Now we can stop there. Okay. Thank you. So it's made us move away from thinking about earlier development of circuits associated with reward, desire and rage before sort of cognitive control circuitry may be forming, to one that may illustrate sort of theories that Thalen and others have suggested in motor development, where there's hierarchical development of circuits. And early on, you get this development in microcircuits. And in fact, I got excited about this because of early optogenetic studies, where you can activate the projection from the basolateral amygdala to the ventral striatum, and you get motivated action. Now, we can't do optogenetics in adolescent mice, but then Kay Tai went on to do some other studies where she was beginning to look at the involvement of the prefrontal cortex and its projections to the amygdala. And thinking about, then we might move across development to more cortical, subcortical regulation and impacts, downregulation, and then these cortical, more complex circuits interacting in later adolescence. But still seeing functionally significant changes during this time. Just to highlight one more area, and that is, there's been work where we've tried to look for signature of brain maturity. And Leah's just got this beautiful paper in Neuroscience, I highly recommend, but she basically says it depends on the imaging modality that you're looking at in terms of when we mature, basically, if that's what you're looking for. And so we have to be cautious. And she also herself has this visceral reaction as a neuroscientist for anybody to talk about maturity, because the brain is plastic and we continue to learn, right? And that suggests kind of stability. But regardless of the imaging modality, you still tend to see this pattern of prefrontal areas tending to show more protracted development. And so we actually took a stab at trying to predict actual age from all neuroanatomical data. This included T1s, T2s, diffusion-weighted imaging. And you can do a pretty good job actually using that information. You can come up with a model that can predict actual age. But the interesting figure in this paper that I love, it's this one. It shows at each age, the contribution of measures that are helping that model predict are very different. And so if I summarize that in words, like their cortical areas is the strongest predictor in childhood, whereas subcortical volume is a stronger predictor in adolescence. So definitely showing this dynamic pattern that is happening. But if we really want to look at the function and maturity or changes in brain during this period, perhaps what we need to do is probe it, and kind of like a cardiologist with a stress test. And since I'm so interested in adolescence, probe it and perturb it into an emotional state, and then see what the pattern is. So this is a collaboration with Damien Fair and Mark Rudolph, where we basically tried to develop a model to predict true age based on functional connectivity data that was either collected during a neutral state or an emotionally rousing state. And so first they trained a model with a subset of the participants in the neutral state, and you see that the model does a pretty good job predicting true age, but that's not what's important. It's whether or not you actually see that this model works with a new independent sample, the test group. And not only do we want to know if that model predicts age in the neutral state, but also in a negative state where we play, I'm not going to play it for you, it's this awful aversive sound. You get a really large galvanic skin response, and the participants hate it, and they rate it as really negative. And then a positive state, where just like with the negative state, they don't know when or if it's going to happen or how loud. In the positive state, we say, we don't know when or if it's going to happen, but at some point in the experiment, you could get $100. And so, and we show them a $100 bill, so that gets them all excited. So this is for Ryan. Yeah. So now let's look and see how that model, how well it predicted. And in the neutral state, it does a pretty good job. But really, the model is much poorer in its performance in these emotional states. So my question, the interesting one is, so where is it failing? Where is it performing poorly? And what does that tell us in terms of the model and development and what emotional states may be doing? And if you plot it, this is looking at whether or not an individual is predicted to be younger than they were or older than they were in these emotional states relative to the neutral state. You see that it was adolescents who were predicted to be younger, and that's where the model performed more poorly. But in this audience, we have astute eyes who are looking at all these dots down below going, but wait, what about all these individuals? And so the question is raised then, if you are predicted to be younger than you are, based on your functional connectome, how does that relate to performance? And since we're talking about adolescence, and we were interested in this with the age range we were evaluating, we wanted to see how it was associated with risk performance. And so I'm going to plot bars that are either open or closed in the field. The open ones are individuals who are predicted to be younger, and the ones that are filled were predicted to be older. And you see this main effect of those individuals who have a less mature functional connectome in these arousing states have higher risk preferences. And you can particularly see this during adolescence, really suggesting that there's a lot of variability in development too. And just because you turn 18 doesn't mean you're magically an adult, even though our legal system suggests that's the age of majority in a number of ways. You can still get the death penalty, but you can't buy alcohol when you engage in behavior at 18. So Allie Cullen, who introduced me, wanted to take this a step further and see how these emotional states actually influence our cognitive abilities. And would we see maybe more protracted development or reaching developmental asymptote later in cognitive abilities if individuals were in an emotional state? And she had positive and negative. I'm just going to focus on the negative ones in the interest of time. So these are showing these fearful faces. And this is a go, no-go task, so they're inhibiting a response to them. And then this is that loud aversive sound that is uncertain, and they don't know when, and they don't know how loud, and they can't do anything to stop it. And what you see is that 18 to 21-year-olds, just like teens, are significantly different in their performance relative to adults. And if we look at neural correlates related to this behavior, we see that these younger ages are not recruiting dorsolateral prefrontal areas as much as adults are, and instead are recruiting areas in medial prefrontal implicated in limbic cortical circuitry. So how has cognitive neuroscience informed our understanding of the developing brain? Well, it's not just simple linear growth or change. We can't just easily superimpose an adult model onto a child or adolescent brain. It really involves dynamic and also nonlinear changes, and they appear to extend into the 20s. So now let's quickly move into what have we learned from cognitive neuroscience in terms of law and policy as first treatment, and then if we have time, I'll talk about diagnostics and targeted treatment. So many youth, unfortunately, come into contact with the US legal system during this extended period of adolescence that you can see. It goes right up until the early 20s. Maps on really nicely the changes that I was just showing you from the Rudolph paper and the Cohen paper. And it's the case that a lot of our developmental science, both in brain and behavior, has informed a number of US Supreme Court rulings, at least those of us in the United States, and actually we've cited work around the world. And so this is just an example of how in the US we have banned the death penalty and mandatory life without parole for individuals under 18. But there's now an APA task force where we're reviewing the neurocognitive evidence in an effort to see what the relevant data are for extending ROPR beyond 18. And there's much more work that I could spend time on, but I just want to switch to thinking about how cognitive neuroscience has informed diagnostic classifications and treatment targets. And then I'll let us go to the poster session. So this is work by two former graduate students, both MD PhDs, Andrew Drysdale and Connor Liston, where they looked at over 1,000 resting state scans, and they were trying to look at different neurotypes or neurophysiological types of depression. They identified 4, but I think what's important to note is none of these biotypes could be differentiated by general clinical features like the Hamilton D. These are very measures that we have used for many years in looking at clinical outcomes of new treatments, using that as the outcome measure. Now, it's not to say that they weren't related to symptoms whatsoever. They were. So we saw that there was one biotype that was related more to anhedonia and another biotype that was related more to anxiety. But you can have these different sort of classifications of subtypes of depression, which is such a broad phenotype anyway, to try to link it to the neurobiology. But what really matters, if you can predict treatment in response to what you've learned from these different biotypes. So in talking with Connor, we had these discussions, and there was a whole subset of individuals who were getting repetitive TMS that were, whose data were in this trial. And so what he examined and showed is that there was one biotype that had a rather big TMS response rate and also showed improvement in depression severity. So this might suggest that novel subtypes like these, we might be moving in the direction of going beyond our current diagnostic boundaries, and they might be useful in us identifying individuals who will be most responsive to these targeted neurostimulation therapies. Now, this was an adult study, and I just want to end with some developmental work, because I think it's important when we're thinking about targeting our treatments to target those to the developing brain as opposed to assuming a developed brain. And so one of the areas of most of our work that's been concentrated is in the area of anxiety and stress-related disorders, in part because there have been really high estimates of this impacting as many as 1 in 4 young people. And very early on, in collaborative work with my postdoc at the time, Katie Thomas, she showed exaggerated amygdala activity in children with anxiety disorder, and she saw that the magnitude in the activity in the amygdala to these fearful pictures was correlated with symptom severity. But I just previously showed you that it may not be about magnitude, but actually the pattern of activity over time with repeated exposure to these cues, that is what is related to anxiety, based on Todd's work. And let's just say, if we're talking about development and we're using faces, we have this huge confound that adults have more experience with faces than adolescents, and adolescents have more experience with faces than children. So we wanted to move away from that and find a neutral, like a neutral cue, and pair that with aversive stimulus, so we're basically talking about cued fear conditioning. And so here we wanted to look at developmental changes in acquisition of cued fear and the extinction of cued fear. And particularly extinction, like once you learn an association, is there a developmental phase in which it gets a little sticky and it's hard to let go? And this is work in humans and mice. With mice, just a few foot shocks paired with a tone, they learn very quickly that the tone is associated with an aversive outcome. And then you can look at that learning by putting them in a novel environment and playing that tone again. And then for extinction, you continue to play that tone without the aversive stimulus. And with humans, with this developmental study, we didn't shock them. We just played that awful aversive noise that they don't like. And we paired that with a colored cue. And first, Siobhan, Stephanie, and Kate Hartley, what they showed is children, adolescents, and adults, you get an aversive stimulus paired with a cue, you learn pretty quickly the cue that is associated with that aversive condition. So we had relatively equivalent acquisition of cued memory. But what about extinction? And what we showed here is that adolescents, both adolescent mice and humans, showed diminished fear extinction. So the fact that the bar is lower, don't be fooled. What it means is humans who had a large conductive stim response just aren't changing very much. They're still showing it, and the mice that were freezing are still freezing more. And you don't see that pattern in the younger animals or older ones, or children or adults. And so if we think about treating anxiety and stress, one of the evidence-based is cognitive behavioral therapy, and some versions of cognitive behavioral therapy include exposure therapy. So you're giving these repeated presentations of triggers that set off the anxiety or stress. So Andrew Drysdale wanted to look at existing data from the New England Journal of Medicine, where there was a clinical trial, and he wanted to see if there was a difference in the efficacy of treatment across development. And what he showed was that there was a non-significant but a diminished response in adolescents in response to the CBT, which was proof of principle because it only had like 2 weeks of exposure therapy, and the other 4 to 6 weeks were other aspects of the cognitive therapy. But these data suggest that there may be a way not to give up on CBT, but we want to boost that up a bit more. In fact, we want to boost it up to all so we get a much higher response. And so the last data that I want to show you is on this slide, and it's how these preclinical data have been associated with efforts by a number of groups to see how we can try to change or help CBT in ways particularly for exposure-based therapies. And so one study is a collaboration with Dave Johnson, and we were really building on the elegant work by Joe Ledoux and Liz Phelps, and showing that when you combine reconsolidation update with extinction and really try to target the memory itself during protein synthesis when it's more malleable, that you can change the memory at the level of the basal lateral amygdala, and you don't have to worry about really requiring the prefrontal cortex to try to resolve the competition between the safety cue during extinction, right? It's not associated with anything, and the threat cue for when it was acquired. And so we've seen success in actually being able to diminish cued fear memory using this technique. Another is to try to engage alternative circuitry by manipulating the context. And so Siobhan Patwell has shown if you put mice back in the chamber in which they were conditioned, you're actually able to see more of diminished cued fear memory with extinction in that context, which I think has several implications and supports some of the work that's being done with virtual reality, where you're trying to get the patient back into that setting where the original trigger began to emerge and solidify. And then the last is Dylan G., a former trainee but a faculty member, colleague at Yale, has also been trying to tap into alternative circuitry using safety signals. So basically, pairing during extinction a cue that has never been associated with any threat. And she, too, is showing that you're getting this diminished, enhanced extinction learning and diminished fear response to the cue. So I hope I've given you a few examples of how the field is changing, and with advances in imaging, analytics, and the access to big data and the use of animal models, how we're enhancing our discoveries that hopefully will inform policies that will change the lives of young people. And by changing their lives, we are ultimately benefiting our own lives and our society. So just to acknowledge the funding, lab members, collaborators, my son, who has given me a unique lens into the extended period of adolescence, and thank you.